Welcome to the DNA fingerprinting simulation. In this simulation, a violent crime has been committed and blood evidence is at a crime scene. You will collect blood samples from the crime scene and use gel electrophoresis to compare DNA samples with DNA samples on file from a suspect DNA profile database. To begin, click on the Sim tab. You will see a crime scene with 6 blood spatters, 6 swabs, and test tubes. Begin by dragging a swab to a blood splatter and releasing it. It will collect a blood sample and deposit itself into the first test tube. Once one blood sample is collected, you may either collect more samples or go to the analysis lab. To go to the analysis lab, click the blue Analysis Lab button on the bottom of the screen. You'll see a lab with a gel electrophoresis tray, a power supply, a pipette, a box of clean pipette tips, a box of used pipette tips, a bottle of gel buffer, and the blood samples. To analyze the blood samples, pour the gel buffer onto the electrophoresis tray. Click on the gel buffer and drag it over to the tray to pour it. Now you're ready to place your blood samples in the tray. Click on the pipette, drag it to the clean tips box, and release it. A clean pipette tip will go onto the pipette, and then you can drag it to a blood sample. Release the pipette over the blood sample and it will collect a sample. Drag the pipette to the gel electrophoresis tray and release it. If you have more than one blood sample to analyze, drag the pipette over to the used tips box and release it so that the tip will be disposed of. Drag the pipette over to the clean tips box and release it, and a clean tip will be placed on the pipette. Drag the pipette over to the next blood sample and release it. Next, drag it to the gel electrophoresis tray and release the sample. Continue until all of your blood samples have been placed on the tray. The pipette will go over to the used tips box and deposit the used pipette tip in the box and return to the tray. Next, you should correctly connect the black and red wires from the power supply to the gel electrophoresis tray. After connecting the wires, click on the switch on the blue power supply box to provide power to the electrophoresis tray. When the electrophoresis is complete, close the message window and click on the DNA results button. You will see the gel electrophoresis tray with purple bands, a ruler, a victim DNA profile database, a suspect DNA profile database, and in the top right-hand corner, the DNA results code. The database positions in the databases refer to the placement of the bands on the electrophoresis tray. To measure the positions, drag the ruler to the tray and line up the 0 on the ruler with the right side of the sample window. You can now measure the placement of the bands and compare them to both the victim and suspect databases for identification.